All right, today we're going to talk about a technology that seriously feels like it's been pulled straight out of a sci-fi movie. It's about a total revolution in how we see things, a way to solve one of the biggest mysteries in medicine by, get this, giving us the power to watch life itself. Think. We're talking about watching cells adapt in real time.
You know, this has to be one of the most heartbreaking questions in all of medicine. A patient is in remission. The scans are clean, there are celebrations, and then months, maybe years later, it's back. And when it comes back, it's almost always more aggressive, more resistant to treatment. So why? What is actually happening in that quiet time between what we thought was a victory and the relapse?
To even begin to answer that, we have to stop thinking about cancer as just a thing, a disease. It's way more clever than that. It's a strategist, and it has a hidden superpower. And that's what we're going to get into right now.
So think about this for a second. Cancer isn't some kind of alien invader. No, it's a civil war. It's started by your own cells. It's your own biology, using the very tools that are meant for survival, like adaptation and evolution, but it's turning them against you. The enemy knows the battlefield perfectly because it is the battlefield.
And that right there, that's its ultimate weapon. It's not about how fast it grows. It's about how fast it learns. You see, treatments like chemotherapy, they're like a huge storm. They wipe out most of the cancer cells, sure, but the ones that survive, well, by definition, they're the ones that had some kind of secret advantage. And then they multiply, creating a brand new, upgraded, resistant version of the tumor. That's the superpower.
So, okay, if adaptation is the real enemy here, why can't we just see it happening? Well, for decades, we've basically been fighting blind. We've been held back by what you could call biology's snapshot problem.
This quote just nails it, doesn't it? I mean, imagine trying to understand an entire football game by just looking at a few still photos. You'd miss everything that matters: the plays, the strategy, the momentum. For so long, to study a cell, we've had to freeze it, stain it, and kill it. We've been taking snapshots. But the whole story of cancer's resistance, it doesn't happen in the snapshot. It happens in the motion, in the movie between those snapshots.
And this is really the core of it all. On one side, you have the old way. It's static, it's destructive, and it's based on averages for millions of cells. The problem with averages is that they can hide that 1 in a million dangerous cell that's going to cause the relapse. But on the other side, that's the dream. A continuous, non-destructive movie of individual living cells. We'd finally be able to stop guessing the story and actually watch it unfold.
So, how does cancer adapt so fast? The answer, it turns out, isn't just in our DNA. Nope. It's in this secret world that actually controls our DNA.
And that world is called epigenetics. Think of it like a master control layer. If your DNA is this massive library with every book your body could possibly need, epigenetics is the librarian who decides which books are open on the table and which ones are locked away in the basement. Cancer is a master hacker of this system. It can flip the switches to turn on survival genes and turn off self-destruct genes, all without ever changing a single letter of the DNA code itself.
I absolutely love this metaphor. The DNA map of your body, your genome, it's pretty much fixed, right? But epigenetics, that's the weather. It's dynamic, it's always changing. It's the rain, the sun, the storms. It's what's happening right now. And cancer just thrives in that chaotic weather, using it to find new paths around all of our best treatments.
So, to beat an enemy that adapts this quickly, we have to be able to see it in real time. We have to solve that snapshot problem. And the solution that's being proposed is just brilliant and incredibly simple at its core. It's to use the oldest, most fundamental messenger in the entire universe: light.
This is the Compass platform from PreciGenetics. And you can think of it like a telescope, but instead of pointing it up at the stars, you point it inward, at the entire universe inside a single living cell. It doesn't use any dyes or labels that could damage the cell. It just uses light, pure and simple. Okay, so how does this actually work? Well, it's this really elegant four-step process. First, a single living cell is guided into this teeny tiny channel. Second, light is passed right through it. Third, the platform reads how that light scatters and changes as it interacts with the cell, and this creates a unique spectral fingerprint. It's like a super detailed biochemical signature. And finally, and this part is absolutely crucial, AI acts as a translator, turning that incredibly complex light signal into a clear, computable stream of data about what the cell is doing right now. This table really just lays it all out, doesn't it? The leap forward is huge.
Traditional methods, they're destructive, they give us those static snapshots, and they rely on these blurry averages. But the Compass platform is non-destructive, it creates a real-time movie, gives us single-cell precision, and here's the real kicker, it shifts the focus from the static genetic code to the dynamic epigenetic control system. It's a whole new way of seeing.
So what happens when we can finally see biology with this kind of clarity? Well, the implications are just staggering. This is where we might see a fundamental shift from just reacting to disease to actually being able to predict it. And to really get a sense of the scale of this challenge, just hold this number in your head, 30 to 40 trillion. That's how many living, communicating, decision-making cells are in your body. We're talking hundreds of times more cells than there are stars in our own galaxy. We are trying to understand a biological universe of just unimaginable complexity.
So, by creating what you could call a living cell atlas with this real-time data, the promise becomes monumental. We could actually predict how a patient's specific cells will react to a drug before they ever take it. We could see the very first flicker of drug resistance as it's happening, not months later when it's too late. This is the big shift, moving medicine from a game of trial and error to a science of measure and predict.
Really, what this all boils down to is that we're standing on the edge of a new era. For centuries, we've been fighting cancer by chasing it. But with tools that let us see its strategies unfolding in real time, we might finally be able to get ahead of it. The goal is no longer just to fight cancer, but to outthink it and outpredict it.
And that kind of leaves us with this one big, final thought. For all of human history, the inner life of our cells has been a complete secret. We could only guess, we could only infer what was going on in there. But what happens to medicine, to us, to humanity, when we can finally pull back that curtain and watch the incredible, dynamic, complex process of life as it thinks?